In eukaryotic gene regulation, we will begin with DNA transcription, which has 3 stages: initiation, elongation, and termination. In the initiation stage, we have transcription factors. Transcription factors are proteins that influence RNA polymerase to transcribe DNA into RNA. 2 types of transcription factors are general and regulatory transcription factors. 35 base pairs from the start line of transcription is the 35 box. General transcription factors will bind to the TATA box. This will influence RNA polymerase to bind to the core promoter. This will cause DNA strand to unzip and have 2 strands, the template strand and the coding strand. Upstream from the core promoter is some control elements. That are going to help control gene expression. Some control elements can be close to the promoter, or they can be farther away upstream to the promoter. Regular proteins are activators and repressor. If an activator binds to DNA, it will increase the rate of transcription. An activator can bind to an enhancer, which is a regulatory element which will increase transcription. If a repressor binds to DNA, it will inhibit transcription. A repressor can bind to a silencer. This will decrease the level of transcription. Another thing that can happen is that an effector molecule can bind to a regulatory transcription factor and promote its binding to DNA. Once an activator is binded to DNA by a zinc finger or a leucine zipper, DNA polymerase is binded to DNA and the strands separate. It will now move into the elongation stage. In the elongation stage, DNA polymerase will read in a 3 to 5 direction, but it will transcribe its complementary strand in a 5 to 3 direction. As we now enter into the termination stage, RNA polymerase will keep running down the template strand and keep making complementary strand until it hits a termination sequence. This will stop transcription. RNA polymerase will release from the template strand and release pre-mRNA. Before translation can happen, pre-mRNA must be processed and converted into mRNA. To convert into mRNA, we must have splicing, a 5-cap, and a poly A tail done. In splicing, this will remove introns, which are these orange pieces, must be removed because it will not translate into proteins. Only exons will, which are these in the green. In order for splicing to occur, a spliceosome has to form. Spliceosomes will bind to the ends of the axons which will split the introns from the pre-mRNA. And it will release into the nucleus to be degraded. To prevent degradation on mRNA, we need to add a 5 cap and a poly A tail. On the 5 end, we will add a cap to protect it from degradation from exonucleases. On the 3 end, we will add a poly A tail, which consists of a group with a lot of adenine nucleotides. Now that we have removed the introns and added a 5 cap poly A tail, we have mRNA that can now leave the nucleus and enter into the cytoplasm for translation.